here's the animation. So what's happening? So we see cells splitting, cells going through phases, the process of how the cell splits. Do you see anything happening inside the cell? What allows it to split, or what has to happen for it to split? What do you think those yellow things are that look like little flies? There you go. There's a keyword, nucleus. So you saw the nucleus disappear. Good. That's a key feature. So we have a nucleus here, and then we saw the nucleus is no longer there. Now we see the nucleus is back. Yellow things are chromosomes. It forms a chromosome panel. That's a good word. So you saw the chromosomes all came together in the center before they separated. Those are some good things to notice. Chromosomes are left behind after the nucleus disappears. Yeah. So what we're going to want to know basically is what the chromosomes are doing and what's happening inside the cell through each of these phases. Okay. So I'm just going to do my agenda again since you didn't get to see it. So we did that. We're going to do the life cycle of the cell notes today. Part of it we will do together, and then you will continue that on your own. So I only posted the student version this time. So maybe split your screen and put the student version up and on one side and have me up on the other side. Tonight you're going to do 2 CK12s. They're due tomorrow by 9. You know, those are relatively short. I have not posted a vocab. Oops. The final exam is the topic list and the review questions are posted in Google Classroom. The questions are very specific to what's on the test. So it should be a very good study tool. It will be overwhelming if you wait to the last minute to do it. So we want to work on it a little bit at a time. Okay. Tomorrow during flex time, we'll do a test correction so you can earn back some points. So if you haven't taken the test yet, I need you to stick around today after the Zoom to complete that test, okay? I'm going to shut my door real quick. So then we just looked at what was posted for today. So we're doing the Zoom. This is due today. That's your student version. Part of it we'll do together, and then the last piece of it you'll do on your own. These two, the next two, these are your CK12 reading with those practice questions. And the last two are videos in CK12. These are not due until Friday. So you have all week, fit them in where you can. Okay? So that was our agenda. All right, any questions on the schedule? So you should have a life cycle of the cell PowerPoint in there, and it's a student version. Remember how to split your screen. You click the tab and pull it out, and you can bring it out all by itself. Okay, so then you can make your screen smaller. So there's the two big phases, interphase and mitosis. Why would a cell even need to go through the cell cycle? First, it might be clearly obvious for unicellular organisms, right? They have a lifespan the same as we do. So they're going to need to divide in order to make more organisms themselves. So kind of like the survival of the species. In order to go from a single cell of an egg to a multi-cell of you, obviously we're going to need to divide one cell and make many cells, because we're made up of trillions of cells. So to create that multicellularity, and also to grow. So you grow by getting bigger, by cells increasing in number and in size. Okay, and then replacement. So our cells themselves have a lifespan, like your red blood cells live for 120 days, and then they die. They get reabsorbed in your spleen, so we have to replace those red blood cells. And repair. So I'm sure all of you have had cuts of some sort, and then we need to repair that cut or a broken bone or a sprained ankle, something like that. All of those injured cells would need to be replaced. Because when you break a bone, the cells that are in that break part, they actually die. So you need to create new cells. Not all cells have the same lifespan. They do go through the same phases of the cell cycle. So like an embryo is dividing every 20 minutes. Like the cells, when you're first forming, they're dividing very, very quickly. That's the rate that a bacteria divides as well. Skin cells, you're losing skin cells every single day. Places where you have a lot of friction in your mouth, in your digestive tract, your skins, you are losing cells daily. So they're replacing themselves daily. So they have a lifespan of only 12, 24 hours. Liver cells, they go into this resting phase and they only divide when they're stressed. So they divide maybe once or twice a year. Some cells in your body don't ever divide. Red blood cells lose their nucleus, so they don't divide. They just die, and we have to make more from stem cells. Nerve cells, you're born with all the nerves you're ever going to have. You don't make new nerve cells. And muscle cells as well. So muscle cells, you're just getting more muscle by increasing the cell itself. Okay. So eukaryotic cells, that's the kind we have, right? Normally I would talk about bacteria, but we're doing the short version and they're not on your exam, so I'm not talking about them. So we have a nuclear membrane we need to deal with. We have all kinds of chromosomes, like we have 46 different chromosomes we have to line up. We have to multiply, we have to divide. Same thing with all the organelles, everything that is inside of our cell, right? Ribosomes, Golgis, mitochondria, we need to make more of all of them. Okay, so all of those are going to need to duplicate all that stuff inside in order to make more cells. So I'm going to play a little animation. This is going to show you what it looks like as the cell divides. So that's a real simple version, right? So you can see the cell creates two nuclei and then it divides. And all of your cells are going through that process. Okay, so this happens over 3 phases. Interphase, often considered a time of rest, but it's not rest. It is a time of cell growth. And in that, we're going to talk about these 3 phases. Mitosis, which is the division of the nucleus itself. And cytoplasm divides, or cytokinesis divides the cytoplasm. Okay, so you divide the nucleus first. So now you have a cell with 2 nuclei, and then you cut the cell in half. And that's cytokinesis, cytoplasm, right? So for the exam, we want to know we spend the majority of our cell's life in interphase. So this is the time between cell divisions. I think that's a question on the exam. So when I say these things, you could highlight them, asterisk them, draw a square around them, maybe write a note in the student note, at the bottom of the slide. So genetic material is in a thread-like form at this point. It's called chromatin. So we talk about chromatin, chromatids, and chromosomes. So at this point, it's chromatin, which under the nucleus or under the microscope, you wouldn't be able to see individual chromosomes. You would just see a mass of nuclear material. Remember DNA and RNA from chapter 6. So we can see G1 is a period of growth. S stands for synthesis. You're synthesizing DNA. So if you want all of your new cells to have the exact same copy of the genetic material you already have, you're going to need to make a copy. So that happens during the S phase. And then G2 is doing some preparation, making materials that's needed for the division itself. Okay, so we can see big portion of the pie is in interphase. So here's those 3 phases. The cell doubles in size in G1. G1 we call the growth phase. That is going to be a test question on your exam. Something like, which phase does the cell do the majority of its growth? That would be G1. The S phase. S stands for synthesis. Remember, synthesis is building. So what are you building? You're building the genetic material. I think that's a test question. The second growth phase is more of a preparation phase. The cell isn't necessarily getting bigger. It's just making materials needed for division. So we'll introduce the term spindle fibers. So spindle fibers are needed. They'll move the chromosomes around. You saw those yellow things in the animation, right? They were moving. So the spindle fibers are what do that. So those are going to be made in the G2 phase. All right. Any questions so far? Tell me to slow down if I need to slow down, okay? So after interphase, we get into the division of the nucleus itself, mitosis. So you have one cell that's going to become two, right? So each of those cells need identical copies of the nucleus. So we need to divide the nucleus in half. So I have happens in most cells because I told you, for example, red blood cells never divide. They lose their nucleus to make more room for hemoglobin and oxygen to carry to your cells. So mitosis is it's a part of the process of cell division. It's specific to the nucleus, and it comes in 4 phases. So you can remember PMAT for short. Prophase, Metaphase, Anaphase, Telophase. PMAT. Okay? So what you really want to know is like key features of each of these phases. So looking at these cells here, this one is going to be interphase. Notice the nucleus is present, and I don't see individual strands of chromosomes. Okay, this is prophase. The chromosomes are visible, and they're still contained in a nuclear region. During metaphase, they line up at the middle. Meta literally means middle. During anaphase, they pull apart. So ana literally means apart. And then this is telophase. Telo is end. So this is your end phase. You can see it's starting to divide the cell itself. Okay? So you want to recognize what are the chromosomes doing in each of these phases. So to talk about this, we need a little vocabulary. Take a minute and see if you can guess where these different structures go. I'll give you a pause before I reveal the answer. So first, see if you can make guesses. You just might be able to figure it out. So here we go. The first obvious one to me are these strings right here. So these are your spindle fibers. So we're going to be talking about these things as we go through the phases, so it's important we know what they are. Centrioles. Centrioles are only present in animal cells, not in plant cells. Okay, but the spindle fibers come from the centrioles, we can see here. Duplicated chromosome. Would we want to put it here or here? I'm going to put it here. This is your original chromatid. No, I'm going to change that. Let me look here for a second. This is a chromatid all by itself. And in a pair, these are sister chromatids. I'm struggling with my words. So this is what's tricky. When it's by itself, it's considered a chromosome. And when it's paired with its identical twin, it's a chromosome together. And within the pair, you have sister chromatids. I think what I want to do is move this. And this is perhaps the duplicate of that. No, I don't like that either. I should have done this first. Sorry, you guys. I'm going to keep it the way I had it. Individually, they are all chromatids. Okay, so when they're by themselves and not in a pair, they're considered a chromosome. So you have 46 chromosomes and they look like this. After you go through interphase and you make copies of them, they appear in pairs. So now this is the duplicated version. And within the pair, you refer to sister chromatid. That part is tricky. What do you think I'm supposed to type here? The phase? I'm not exactly sure. Okay. So that's the way I would line this up, and we're going to keep going. So what I just want you to know what these different structures are as we talk about them, okay? And we'll have pictures. So in prophase, a couple things are going to happen. So the DNA begins to, remember we said it was in chromatin and you couldn't see it. So it's going to coil up, so it wraps up and it becomes very thick, which makes it visible. At this point, we're going to refer to them as chromosomes, okay? Spindle fibers extend from the centrioles here. Notice the spindle fibers are beginning to form. They will attach to the chromosome at the centromere. So the centromere, this little structure here, this is where the spindle fibers will attach, okay? By the end of prophase, the nuclear membrane will have broken apart and get stored in vesicles. Remember those little baggies? So they're going to store the vesicle, store the nuclear envelope so that they can form it again later. So this is what it would look like. So remember, interphase, you don't see individual chromosomes. They begin to thicken up. So to me, this looks like a polka-dotted nucleus. So they begin to thicken up at early prophase. They're still in a nuclear region. By late prophase, by the end of prophase, that nucleus has disappeared, okay? This is showing you how the chromatin gets coiled up into chromosomes. So metaphase, we think meta meets at the middle. So meta means middle. So the chromosomes at this point are all lined up at the equator. The chromosomes are made of sister chromatids, two identical copies. So like in this cell, you have 4. Let's call them 1, 2, 3, 4. So this is 2 copies of the first chromosome, 2 copies of the second, 2 copies of the third. And within the pair, you refer to the sister. So you have the identical, you have the original, and then you make a copy, and it's the sister chromatid. Okay, so they line up at the middle. This is what it would look like under a microscope. Metaphase is the easiest phase to identify. Anaphase, the chromosomes will pull apart. So sister chromatids separate, and they move to opposite poles, the ends of the nucleus. I'm sorry, the ends of the cell. Okay, so you have this chromosome. It made a copy and paired with its sister. And once pulled apart, each of these are once again considered chromosomes. Okay? Because that chromatid is more of a relationship. During telophase, everything that happened in prophase unhappens. So the chromosomes will uncoil, become chromatin again. The nuclear membrane is going to reform. So spindle fibers disappear. The vesicles, they all come together. Remember, they were storing components of the nuclear membrane. So they're going to come together and reform that nuclear membrane. And then everything uncoils and becomes thin again. So in the end, each daughter cell has the exact copy of the original parent cell. So if a cell has 12 chromosomes to begin with, how many will each daughter cell have? Will they have half, or will they have a whole set? It starts with 12. Does each daughter cell end with 12, or does each daughter cell end with 6? What do you think? Notice my example. I've been using 4 chromosomes through this whole thing. 4 chromosome splits. 2 daughter cells each have 4. Okay, So this is an asexual reproduction. It makes clones, okay? So it makes an identical copy of the original cell, which means it has the exact same number. That is an exam question. Given a number, if a cell has 24 chromosomes and it goes through mitosis, how many chromosomes will the daughter cells have? It starts with 24, it ends with shout or put it in the box. There you go. Good job. See, that's an exam question. You got it right. I use a different number though. Maybe I say 18 and it ends with 18. Maybe it's 12 and it ends with 12. The point is, it ends with the exact same number. Okay? So that's an exam question. Cytokinesis, I'm going to play this out. What you're going to see happen in cytokinesis, the nucleus has already divided. Now we're dividing the cytoplasm, making two cells. Okay?
New cells are formed by cell division, in which one cell splits to become two cells. Cell division involves two processes. In the first, called nuclear division, the nucleus divides. In the second process, cytokinesis, the cytoplasm divides, producing two cells.
You see how it gets pinched in half, right? So you refer to that as the cleavage furrow. So animal cells and plant cells are a little bit different because the plant cell has a cell wall. So with animal cells, pretty simple. This is a fish blastula. You just have a cleavage furrow that divides. It's kind of like if you had some slime, you know, and you pinched it in the middle, you'd be able to pull it apart. Okay, so just pinching it in the middle. Plant cells have to form that cell wall. So they create a cell plate with vesicles. Remember, vesicles store things. And then the vesicles will release the cellulose on either side. So from the cell plate, they create a cell wall on both sides. So if you saw a cell plate or a cell wall under a microscope, you would know that you're looking at a plant cell. So this is showing you the vesicles coming together in the middle, forming the cell plates. Oops, I go down here, cell plates. And then they release their contents on either side, forming the walls. Okay. At that point, you have two new cells. You've split the nucleus through mitosis. You split the cytoplasm through cytokinesis. Okay. So we're just going to go through a couple of these next slides together. See if you can identify the slides in which phase they are before I turn you loose. Okay. So we're almost done. Anaphase, the key. Apart. Ana, apart. Chromosomes are being pulled apart. Meta, middle. They're lined up at the middle. Interphase, you can't see anything really happening, right? Prophase, polka dotted nucleus, or chromosomes in no particular pattern. They're paired, but they're not arranged. And then telophase, you should have two sets of chromosomes, okay? So take a minute and go through the next handful of slides where all I'm asking you to do is identify the phase. This is the last thing we'll do together, and then I will turn you loose. So I'm going to go through these. This one is anaphase. Are you on the same side as me? The first one, anaphase. I see the chromosomes are being pulled apart. This one is interphase. I see a nucleus. I don't see anything specific happening inside. This is telophase. It looks like two twin cells or one cell that is like been split in half, right? It's almost as big as a full cell. This would be an early prophase. The nucleus, see the edges starting to break apart. I start to see spaces in between. This is clearly an interphase. I can see the nucleus. I can even see the nucleolus. chromosomes are here. This is metaphase. They are lined up at the equator or lined up in the middle. Meta, meet in the middle, MMM. Okay. Again, two twin cells, two nuclei. This is going to be telophase. Two twin nuclei is what I see there. They're still kind of connected, see here in the middle. This is metaphase, lined up at the equator. So you have like onion root tip cells, and the other ones are fish blastula cells. That's why they look a little bit different. So these ones are the plant cells, and the others are the animal cells. So this is metaphase, lined up at the middle. Our nucleus is beginning to really uncondense, if you will. So you might have called this telophase, or you might have called this cytokinesis. This one looks a little further along than the last one we looked at that we called telophase. I can see the cleavage furrow is formed here. Again, a prophase. The chromatin seems to have thickened up into chunks, right? So those are individual chromosomes that we're starting to see. And anaphase. To me, metaphase and anaphase are the easiest to identify. Anaphase, these chromosomes are being pulled apart towards the center. There's no division yet occurring towards the center. Towards the poles is what I meant to say. And that is the last one. So now I just have kind of a checking for understanding. What happened to this slide? Hopefully on yours, you actually have a picture. So you just have 1, 2, 3, 4, 5 slides to do and turn in today. So this is the checking for understanding piece. And that's all I need you to turn in today. And then you have videos by the end of the week and reading by tomorrow. Okay, so what questions do you have for me?